The lipid bilayer is found in the cell membrane, which consists of two opposed layers of lipids. These two layers are to help make barriers on both the inside as well as the outside of the cell. The membrane is known to be amphibiotic, which means it contains both hydrophilic layer as well as a hydrophobic layer. Hydrophobic tails of the layer keep water out of the parts of the membrane, as for the hydrophilic heads, like water, so they are normally don't have a problem with being exposed to liquids. These heads are facing outwards of the membrane, forming a circle, as for the tails being on the inside of the membrane and coming close together. Here, it's shown inside of the cell that, so on top shown right here are the hydrophilic, which means that they're water loving, and then here on the inside of the cell are the hydrophobic, which is to keep the water out of the cell. The tails of the bilayer are known to have kinks due to the double bonds of the hydrocarbon tail, which are saturated with protons. Stating right here shows a kink inside of one of the hydrophilic tails. This allows less density on the layer and helps the system as well. The hydrophobic tails are lined from tail to tail, allowing the heads to face forward, in which the heads are found on both the inside but also the outside of the membrane, allowing a hollow ball to be made. This ball is known to be the liposome, which allow the membrane to be self-sealing. If the cell here is exposed to water, the lipid bilayer must roll into a hollow ball, causing conservation of energy. Now the bilayer is known to be 2D, showing the movement of the membrane to be lateral diffusion, meaning that the layer moves forward, backward, and also side to side. The main function of this membrane is to carry out membrane proteins across the membrane, as well as an anchor protein inside the membrane. For example, bacteriohodapsin protein is found in archaean species called Halobacterium, which lives in very acidic environments. Each of these proteins are found to hold a non-protein molecule, which is known to be retinol. Retinol will absorb a light photon, causing the protein to change in shape to make another proton. For example, in this photo, retinol shown in blue, which is right here, is at its normal state, and once sunlight hits this is when the protein will begin to change shape. And this is caused by an active transport, which causes a molecule, which is also a photon, to move against the concentration gradient from a lower concentration region to a higher one. This transportation needs energy, which is energy is being absorbed from the sun. This will then keep repeating, which the bacteriohodapsin is known to be a transport protein, making photons move across the membrane in and out, which shown in this diagram that retinol had changed shape due to sunlight, and now the hydrogen has moved inside. Once the protons absorb enough, that's when the concentration gradient will go right back to its original shape, and the protons are then forced out by using a pump, which also lowers the pH. All ions and charged molecules, when looking at the charges, require transporter and channels. These membrane transport systems are very exclusive to the proteins that are being brought through the channels and transporters. Channels don't require any energy due to them opening and closing. They go from here, shown in this photo, from high concentration to low concentration using a channel. Transporters, on the other hand, require lots of ATP due to having to bring molecules from a lower concentration to a higher concentration. By the molecules going against their gradient, it is known as active transport, which is shown here in this diagram right here. The movement of these forces going from concentration dependent to other charge dependent is called electrochemical gradient. The main strategies that achieve active transport across these electrochemical gradients are coupled transporters, ATP driven pumps, and light driven pumps. Coupled transporters are one molecule to move with the gradient power of another against the gradient. ATP driven pumps use the energy obtained from hydrolysis of ATP to force movement upon molecules against its gradient. And as for the light driven pumps, is what was described earlier in bacteriorhodopsin. Transporter proteins contain moving parts and transfers molecules from one side of the membrane to the other. Then there is channel, which is a protein with no moving parts. Concentrations on this membrane are very important since it differs from the inside the cell and the outside of the cell. For example, when looking at sodium potassium, sodium likes to be outside of the cell where it is found to have a higher concentration. As for potassium, it likes to be inside the cell due to being attracted to negative charges of the Cl or chloride, which is why higher concentrations will be found there. To neutralize a cell so it doesn't take a specific charge when sodium is moving in, both of the concentrations have to be equal on the inside and the outside of the cell. By doing this, the potassium will be moved through the channel, which is shown in blue, when the concentration of the inside of the cell becomes too positive. Also, small nonpolar molecules are known to be easily diffused across the membrane by themselves without the help of channeling or transporting. Uncharged polar molecules are required, transporters and channel molecules, due to the molecule being bigger. So in this case, when sodium is being pumped into the concentration of the cell, and it begins to be shown with the potassium. The potassium will begin to channel out, causing its concentration to start going to the left side. Once enough sodium is inside the cell, water will also begin to follow. So wherever sodium goes, that's where water will go. And if too much water gets into the cell, potassium will then come back into the cell, and sodium will have no choice to be pumped out using ATP. What makes the cell membrane have supportive framework is the cell cortex, which is made up of a meshwork of proteins that lay right underneath and connect the cell membrane. In this membrane as well, the movement of the proteins need to be very regulated by specialized regions called membrane domain. This allows the membrane proteins to stick to something that lies outside, proteins to stick inside, or structures in or outside of the membrane that restricts the protein to move or float past the region providing structural support so that the cell will not collapse. In eukaryotic cells, their membrane organelles are closed off, not allowing anything to diffuse into or out of the cell without going through the nuclear envelope. This double membrane is made of two bilayers, and the only way molecules could go in and out is through nuclear pores. The nuclear envelope is connected to the endoplasmic reticulum, in which there's a rough ER and a smooth ER. The difference between the two is that the rough ER contains ribosomes and the smooth ER does not. As proteins go through the ER, the Golgi apparatus will begin to accept the proteins and lipids so it could modify and send them out to other or final destinations within the cell. This will be sent out through vesicles called endosomes, which will send back out of the cell and to others in the lysosome. Lysosomes digest engulfed nutrient material, which consume and degrade aged organelles. When looking at why these organelles work the way they do inside the cell today is due to how they worked when they began as archaea. In the early cell, the plasma membrane was responsible for all membrane-dependent cellular processes. As cells began to get more complex, however, the membrane needed to become more sufficient. The intracellular membrane, which included the nucleus, ER, Golgi, lysosome, and endosome, became the endomembrane system due to the organelles always communicating with one another and transferring proteins. Here, when looking at this diagram, you can see how the lysosome, Golgi, ER, and nucleus became one cell. So if you noticed in all four of them, they all started as just a regular cell with nothing from the outside environment. When looking closely to the lysosome, it is noticed that the outside environment goes right inside. As for the Golgi as well, the outside environment makes up the interior regions. And this keeps prolonging and prolonging with the Golgi due to having multiple parts. For the ER as well, it grabs the outside environment and brings it into the cell. The only difference is that the nucleus has both environmental and outside environment that makes up this cell. As for evolutionary organelles like the mitochondria and chloroplast, these still process their own proteins and have no traffic leading to other organelles in the cell due to being independent in the past time. When proteins enter from the cytoplasm, those that don't need to go through the ER go to the Golgi until they go to their final destination. These free ribosomes contain amino acids and have labels called sorting signals. No matter what the final destination may be for these proteins, it has to get across the organelle membrane, in which nuclear proteins enter the nucleus through a portal. Then the proteins will move through the ER, mitochondria, and chloroplast through protein transporters, which then finally the proteins will leave the ER and be traded to other parts of the endomembrane by transport vesicles. Transport vesicles are found to be very soluble and are known to carry these soluble proteins across the membrane with a highly organized system. One system is known as the secretory system, which transports proteins from the rough ER to the outside of the cell. Another pathway is the endosic, which here extracellular molecules are brought into the cell and degraded by lysosomes. Here's where vesicle budding takes place that involves transferring the correct proteins along the right organelles. The vesicle being coated helps to form the bud as well as hold its shape to be able to target the vesicles and being able to transfer the correct contents needed for the proper organelle. The coat of the vesicle is made up of a protein called clathrin, which buds off the Golgi for secretion and will bud off the membrane for endocytosis. Here in this diagram, clathrin is shown in red, adaptins are shown in green, and the cargo receptors are shown in blue. When budding off the cell membrane, each vesicle will start with clathrin, coated pit, and which helps shape the pit to make a vesicle from containing a cargo receptor with their proteins, rabs and snares. Dynamin then comes and will form a ring around the vesicle to pinch left over membrane, releasing the membrane. Here, the dynamin is shown right above in blue, and clathrin is on the bottom, adaptins is the green, and snaps and rabs is found in orange. These signals will begin to attach to the cargo receptors so that the other proteins could be transported to where they're needed. However, adaptins only bind to receptors for proteins that only belong in the cell membrane. So once they react to the cell membrane, it will only contain proteins for that organ. Once a vesicle reaches the membrane, shown here, this is where it buds off, and now it is being seen near the cell membrane. It will only contain proteins for that organ. Once a vesicle reaches the membrane, it will dock, making the membrane to fuse. Rab proteins are the clues on the surface of the vesicle that reach and bound by tethering proteins. So rabs here are found on top, in which tethering proteins, shown as I'm drawing, will be found here. Each of these rab proteins are the clues of the surface of the vesicle that recognize and bound by tethering proteins. Each rab protein has a specific point of origin. Once the protein and the tethering begin, snare proteins will start to stabilize and dock, which is shown on the bottom right here. Now this will cause the membrane to fuse and target. now allowing the membrane to send the appropriate cellular signals. Snares will start to twist together due to the vesicles and target membranes coming very close to one another during membrane fusion. Membrane secretion now has to occur, allowing the new materials needed in the cell to be delivered directly to the cell membrane, which is now shown over on this side. Membrane secretion now has to occur, allowing new materials needed in the cell to be delivered directly to the cell membrane and extracellular spaces by the vesicles known as exocytosis. As these materials are being transferred, they are also being checked throughout the process consistently, checking for damaged proteins, which is then sent back up to the chain or being destroyed or recycled through the cell. Shown in this diagram, proteins enter the ER, sugars and proteins are now added, known as glycosylation. This happens in the ER lumen, where sugar trees is added to the glycoproteins, causing many different trees to be made. As these proteins are made, they soon have to leave the ER. With this, they have to go through a check before leaving. Proteins that are folded correctly to leave the ER are binded by chaperone proteins. If chaperone proteins stay on the folding protein for too long, the cell will automatically begin destroying it. Once the process of destroying happens, it could cause the cell to sometimes undergo cystic fibrosis. However, if the chaperone protein unfolds the folding protein, making it go back to its original shape, it could leave the ER. Once the protein leaves the ER, it then enters the Golgi process, which is now being shown in this diagram, going from the nucleus, the ER, and now specifically to the Golgi. Proteins here will go into a cis phase, accepts the vesicles from the ER, and then will go into the trans phase, which vesicles leave the Golgi. Proteins will go into the cis phase, into the Golgi, to the cisterna of the Golgi, to the medial cisterna, then the trans cisterna, and then finally to the trans Golgi, where the proteins network out of the Golgi, taking the proteins needed with them. As these proteins are being secreted from the Golgi to the pathway, this will allow constant supplies of new membrane lipids and new membrane proteins to the membrane by consecutive exocytosis pathway. However, if the secreted proteins remain as spherical proteins of the membrane, then they begin to be a part of the regulated exocellular matrix. There is also another pathway called regulated exocytosis, which operates only in specialized cells which have been secreted from specialized molecules. The cytoskeleton is made up of 3 different kinds of protein filaments, which are intermediate filaments, microtubules, and actin filaments. Intermediate filaments are the most durable and are known to be the strongest filaments. They are found in the cytoplasm, where a network is formed around the nucleus and extends to the cell membrane. The IFs are made like a rope, where individual fibers and individual strands twist together, providing a lot of strength. The pairs of IFs wrap around to form a dimer, which 2 of them will come together and make a trimer. IFs are mostly found in the cells that experience a lot of stress. They are able to distribute the forces of stress throughout the cell rather than in just one location. There are 4 main classes of IFs, which is keratin, filament, neurofilaments, and nuclear lamins. Keratin is a mixture of different subunits, making it the most diverse IF. Vitamin is found to be the support cells found in the muscle and tissue cells. Neurofilaments provide structural support of neurons as well as neurogeneration in the nervous system. Finally, the nuclear lamins are involved in the strengthening in the nuclear envelope, only found in animal cells. Lamins are very important to the cell division, so without lamin being present, cells will die due to no reassemble or disassemble. Microtubules are hollow tubes of protein that can rapidly disassemble at one location but reassemble at another. They have a place of origin, which are the centrosome, and extend to the interior face of the cell membrane, which causes protein tracks throughout the cell. They are also used as anchor proteins in localized any organelle. They are known to be so dynamic that some begin to be static. Some are found to be outside the eukaryotic cells, found in flagella and cilia. Microtubules are made up of subunit proteins known as tubulin that are made up of the globular proteins. These proteins are made by dimers stacking on top of one another, interacting with non-covalent forces, forming the walls of the MT. Profilaments interact side by side to make a single MT, which each dimer consists of subunits. They consist of beginning and an end on the profilaments. These tubules help ship molecules during cellular transport using polarity. Centrosomes give the tubulin organization, causing tubulin to join constantly, which form the tube, making a ring-like structure made up of another tubulin besides alpha and a beta. The negative end of the MT is embedded into the centrosome, and the positive end is at the growing end, which is shown here in this diagram. Sometimes the MT is known to lose some alpha-beta dimers, causing it to either fall apart a little or to fall all the way. This is known as dynamic stability, in which the GTP binding dimers contains a cap, which is shown here, and plays a role in why these fibers begin to fall apart. Polymerization could slow down the MT, causing the GTP dimers to fray. Once this begins, the GDP dimers break apart, causing the GTP dimers to be exposed, and the MT fibers will begin to be destroyed. This disassembles the fibers and lets the free dimers begin to rejoin other dimers, allowing polymerization to occur again. This process could be stopped if anchoring occurs due to the centrosome unstable, sending out more MT. So if one catches onto a good target, it becomes stable and will not fray. If not, the MT will begin to fray completely. MT helps guide vesicle transport in a uniform direction using polarity as well as a motor protein. These proteins are ATP traveled along the MT in one direction. Kinesins and diamonds travel along the MT as well, with kinesin towards the centrosome on the negative end and the diamond moving away from the centrosome on the positive end. That's shown in this diagram here, where the GDTP cap is on the right, which is going towards the positive end, and the GDP is on the left, which is on the negative end. This all cycles by binding to the GDP, cleaving GTP, and releasing the GDP, almost like the motor proteins are walking. This allows the kinesins to interact with the ER and Golgi by stretching them out. This causes the ER to shrink to the center of the cell, and the Golgi fragments will begin to disperse throughout the cell through the vesicles. Once the chemical is removed, the MT will reassemble, and the motor proteins will cause the organelles to go back to its original shape. As anchoring proteins will not connect to the cap causing GTP to stay in place, the GTP will soon turn into GDP. As diamond walks towards the positive end and kinesin walks towards the negative end, diamond will always become first than kinesin. And the plus end is always known as the retrograde motion, which is going towards the left, and the anterior grade motion is diamond going towards the right. Actin filaments are twisted chains of globular actin subunits that contain polarity. They have positive and negative ends. They are known to be hollow. They are very short, thin, and flexible compared to MTs. Proteins have to be bound to these filaments in order to be stabilized so that they don't disassemble. Each of the actin monomers are bound to ATP and is cleaved to ADP. This causes reduction of the binding of the monomer, promoting depolymerization. Small proteins in the cytosol bind to the actin monomers and block them from joining any growing actin filaments. However, actin-binding proteins bind to already polymerized filaments. These binding proteins hold the filaments in bundles, meshwork of the cell cortex for server any proteins, or they form tracks that could bind to the motor proteins. During the binding process, the cell cortex is known to be the stability of the cell, allowing actin filaments to link into protein meshwork. This allows the cell to change shape as well as move, shown here on a side view and also shown here at the bottom as a top view. During the binding process, the cell cortex is known to be the stability of the cell, allowing the actin filaments to link into protein meshwork. This allows the cell to change shape as well as move. In order for the cell to crawl, actin is used when the cell senses the environmental signals. First, the cell will begin to push out the pododia at the leading edge, and then the pododia will then go to the extracellular surface where the cell will begin to crawl. The cell will begin to flow forwarding to the anchoring points, filling the front of the cell, which is shown in green on the top view. That allows it to move. To allow actin polymerization to occur, lamelliphodia and filopodia will begin to help the cell crawl along the surface they were able to anchor onto. And the lampodia is at the beginning right here, and the filopodia is the little fingers at the bottom. Integrins are the reasoning on why the cell will anchor and begin to move along the surface of the cell membrane. The cell begins to rearrange its cytoskeleton due to the outer responses leading to RHO, small gene proteins that activate when GTP is present and to also be active. This causes the actin polymerization and bundling for the filopodia to form, then triggers the lampodia formation, and finally makes the cell crawl. The outside cues begin to change shape of the cell due to the affecting RHO factor. Cell signaling is a huge communication that happens throughout the cell. Signal transduction allows the cell to signal from one form to another. The cell that sends this information is known as the signal molecule, which does not enter the cell directly, and it normally releases a signaling molecule. This molecule is then detected by a receptor on a target cell that receives information. With all these signals needed to be transduced throughout the body, there are different styles of communicating. The first is the endocrine system, which is publicly a signal that releases hormones throughout the whole cellular system. Then there's the pancrean system, which is less public and releases signals only to the neighboring cells and not the bloodstream. Lastly, there is the nervous system that is directly signal that sends its signal to single destinations. Cells know to ignore signals that aren't meant to be in specific locations so that the destination of the cells know to ignore the signals that aren't meant to be in specific locations so that the cell doesn't have to overload of information coming in and out. The signal receptor is made for a single signal. Sometimes signal molecules are too big to enter a cell during the fusion. In this case, the signal molecule has to bind to a surface receptor, in which the signal molecule will then change shape and become a primary messenger. Signal transduction will then occur, carrying the signal into the cell, which then a second messenger will relay the message again throughout the inside of the cell. This is known as signaling cascade, allowing a signal to be received, turning multiple proteins on, to then signal another molecule and allow more proteins to be on again. However, sometimes modulation occurs in which some proteins try to turn off a message, tweaking it to a place, but the signal could still be found in another. How these cell signals turn on and off are based on the receptors and internal responses. The cell surface receptors have three main categories, in which the first is the ion channel receptor. This allows the receptor to bind to the appropriate signal molecule and opens, allowing ions in. Then there's the G protein receptor, which carries out a signal to the cell by multiple modified proteins. Lastly, there is the enzyme coupled receptor, which the receptor is also an enzyme and is bound to the signal molecule and changes the target molecule completely. G protein coupled receptors play a role in every system of cell communication. These proteins are found at the transmembrane of the cell surface and are synthesized at the rough ER, usually have starting and stop transfer sequences. They are then shipped to the Golgi, processed, and shipped to the cell membrane using transport vesicles. Once bound by a signaling molecule, GPCR causes shape change to an alpha subunit to let go of ADP and take up. This causes the protein to be active and making the signal turn off in the process by allowing the system to be on. G proteins activate the membrane enzyme and channels, as well as opening the ion channels. GPCRs activate adylate cyclase and phospholase C enzymes, in which they indirectly are which indirectly converted into CAMP, which carries messages into the cell, but only does so because it only is a signaling molecule of a message to just be sent. In this diagram, the G protein is bound to the GDP, and currently it is off. Then it will go to this diagram in which now it is now bound to GDP. Which then the protein will then be transferred to the alpha, which will then bind to the AC, and which will then be made into cyclic AMP. And then from there, the cyclic AMP will then bind to the PCA, which is shown in orange, and then it will disperse out into the cell, making multiple proteins turn on. Once cyclic AMP activates kinase A, which causes phosphorylation to turn on or off, a second messenger that is involved with the G proteins is calcium. This is known as the PIP2 that has the G protein activate phospholase C. This splits into two molecules known as IP3 and DAG. IP3 goes into the ER, causing calcium to release, and then will bind to PKC, making it more active, known as calmodulin. Also, enzyme coupled receptors are a little different than GPCRs due to including a kinase as part of the structure. The movement of the subunits in these enzymes will determine the pathway signals of the molecule. Cells undergo a cell cycle in which some cases would be seen with the naked eye and sometimes it cannot. During mitosis, it has been noticed that the cell growth could be seen with the naked eye. There are different phases within mitosis, starting with the interphase. Interphase is the beginning of the cell growth, which includes three phases. The S phase, in which the DNA is replicated. Then there's the G1 phase, is when cells get ready to replicate between the M and S phase. Then lastly is the G2 phase, which is the gap between S phase and the next M phase. During the transferring between the different phases, there are checkpoints in which the cell would determine if the cell could go on to the next phase of replication. This cell cycle control system monitors the network of proteins it begins to signal the cell to get ready to divide. Cells cannot proceed to the next phase without being looked at carefully when going through the system. There are three main checkpoints in the cell, which the first is the G1 phase, will confirm the cellular environment. This is the most crucial phase due to it influencing external signals and cues. Then in G2 phase, it will begin to confirm the completion of DNA replication and repair, which will follow with the M phase that confirms the mitotic spindles attached to the sister chromatids. If none of these phases go until M phase and it stops at G1, this will have the cell go straight to the last phase, which is called G0. This is where cells will not divide but will always stay in place and not move. However, if the cell doesn't go through all of the checkpoints, it could lead to a long-term complications like cancer. During the changes of these checkpoints, each stage contains its own key proteins which are turned on or off. Cyclins control kinases and phosphates during cyclic regulation in a cell. Cyclin will turn a protein into the kinase for that particular action, which will then turn into the proper protein or CDK, causing the protein to be on during the cycle. If the cell does not detect any damage to the DNA, this is where the DNA will begin to check the damage checkpoints in both G1 and S phase. The best way to check the DNA damage is the beginning during G1 phase so that the replication doesn't occur anymore and a bad DNA starts to be made. P53 protein is found to be a transcription factor that activates a CDK inhibitor called P21. This prevents S phase from occurring when the DNA is going through repair, and if it cannot repair, it will then undergo apoptosis.
Without P53, P21 will never be made, causing no cell repair, and without P21, the cell will never get signaled that DNA is damaged. If there is a mutation found on P53, this could correlate with cancer due to P53 not being able to fix the mutations that were found earlier. However, if the cell does repair the DNA, it then continues on throughout the cell cycle.
In animals and plant cells, they contain an extracellular matrix that both differ from one another. In a plant cell, the ECM is strong and durable, just doesn't get to that extent very quickly. For animals, they need to be strong, durable, and flexible very quickly in order to get the strength needed throughout the body. Animals' connective tissue have different ECMs due to each one having a different role or size in each of the systems in the body. There are 4 different categories in which there are connective tissue, epithelial, nervous, and muscular tissue that all contain and have different functions. This is due to each of the different ECM fibrosis proteins containing a different collagen, which is the main structural bearing component in the ECM. Collagen is known to be very tensile in animals' connective tissue. They are winded up by a collagen subunits, creating a triple helix, and are assembled in to be fibrils. Each of these connective tissue collagens have special secreting cells to go along with the importance of which system they are found in. Some of these cells, however, do need to break down and be able to cut through the ECM to allow repair and growth to the tissue, which is known as the matrix prothesis. During secretion, collagen will make the ECM first, and then depending on the environment, it will begin to arrange into layers, allowing either stretching or distortion to occur. Now, on the surface of the ECMs, cell crawling occurs due to actin binding to integrin, integrin binding to fibronectin, then fibronectin linking to collagen fibers, allowing the cell crawling to occur. During this process, cells have to withstand any pressure on the cell, which gags play a huge role by binding to water molecules to form a gel. This gel will swell with water, allowing a cushion to withstand any pressures. Most cells found throughout the body are found in sheets, and each one will have a specific shape depending on the type of tissue it may be. Epithelial cells are known to be the only cells that line the outside and inside of the body. Their function is to keep something in as well as something out. These sheets of cells contain 2 sides known as the apical and basal surfaces. These surfaces are shown here in this first diagram. Apical is on the top and basal is on the bottom, and collagen is found on the bottom as well, which is shown right here. These cells are known to be polarized and are known to have 2 different ends that do different things. Epithelial cells have different ways to connect to other things in the body known as cell junctions. The first junction is going to be shown here on your right. This is an adherent junction, which has actin filaments of the cytoskeleton coverage on the cadherin transmission membrane. Then the cadherin of one cell bridges to the cadherin of another, essentially connecting 2 cells' cytoskeletons through actin. The transmembrane bridges together use calcium. Cadherin is the protein that bridges them, and actin filaments are involved in the cytoskeleton component. This allows movement for the cell. This is the desmosomes, which have IFs of the cytoskeleton, specifically keratin, coverage on cadherin transmembrane proteins. Then the cadherin of one cell bridges to the cadherin of another, essentially connecting two cell cytoskeletons through keratin in an IF. These intermediate filaments connect to stop the tissues from tearing. And then the desmosome allows stretching as well. This diagram is the hemidesmosomes, which also have IFs involved, which is right here on the top. But this time, instead of converging on cadherin, they link to integrin. These integrin proteins then bind to collagen of the ECM, anchoring cells to the ECM by linking it to the IFs of the cell's cytoskeleton. Integrin indirectly connects the collagen, which is shown here on the bottom, causing an anchor to occur, which is right here with the integrins. These cells join together and join cells to another organism. This is a tight junction, which uses a Claudin, which is shown here, and Claudin, which is shown in red here, proteins to zip 2 cells up together, creating a watertight seal, which is shown right here, between them. These junctions are used for
epithelial sheets. And lastly shown here is a gap junction, which involves two cells expressing connexon channels. These channels of each cell line up and connect, creating an open passageway between the two cells through which molecules can pass.
Humans contain trillions of cells that divide rapidly every second. They undergo in the cell cycle in which they are dividing as well as being looked over for mistakes. However, sometimes on our cells, they do make a mistake and it sometimes isn't fixed, in which this case becomes a huge problem. This begins to cause the cells to divide the wrong way and begin to destroy tissues that happen to be in their way, which is known as cancer. There are 3 different tumors that can be caused by cancer. The first is a benign tumor that rapidly grows a mass of cells that usually remains local and can be fully removed by surgery. Then there's a malignant tumor that could invade the surrounding tissues. Lastly, there's a metastatic tumor, which is the most frightening of them all, because not only does the tumor invade neighboring tissues, but it begins to migrate into the blood and lymph supply. Wherever these cells land and divide, a secondary tumor will then begin to retain. When looking at the secondary tumors, they could determine where they originally come from just by looking at how the cells look as well as they act and where they are found other than the primary location. Epidemiology is used to analyze the relationships between certain environment factors as well as human diseases and why these tumors begin in the first place. Environmental effects have a huge role on why cancer begins in the body. STDs and smoking have found to have a huge factor in one's body, as well as other life choices, like having a poor diet. Another way cancer is developed in genetic influences due to the mutations of a somatic cell. Genetic instability has the biggest cancer risk due to the mutation affecting the replication and maintenance of the genome. These mutations cause the DNA to deactivate the repair enzymes of the DNA proofreading, which causes more mutations and increases cancer. It could also bypass the cell cycle checkpoints like p53, in which the cell won't arrest but will continue to divide. Lastly, it could also defect in the machinery needed for the mitosis. Once these mutations begin to occur in the cancer cells, will no longer require signals from another cell to thrive. They also do not go through apoptosis, which is very scary due to the fact that normal cells begin to die off once they know they cannot be fixed. Cancer cells are known to never cease division and are known to be genetically unstable. Also, cancer cells lack cadherin and other proteins needed for cell junctions. Lastly, cancer cells are able to survive and divide in foreign tissues of other cell types. 2 classes of gene mutations known as oncogenes and tumor suppressor genes. Oncogenes are mutated genes that become hyperactive and could cause cancer. As for tumor suppressor genes, they are mutated and become inactive, causing the gene to go from tumor suppressors and being forced to be turned off, which causes cancer. So in an oncogene, cancer is known to be dominant. So in order for a cell to be healthy, the oncogenes must be turned off. Here, both genes are off. If a mutation occurs on one allele, the cancer protein is made, making the cells to signal cancer. So here on the dad allele, which is shown on the left, the big A is shown as homozygous, which is turning the allele on due to a mutation that occurred. This is the mutation. If the mutation occurs, it causes the protein to be made for cancer. And since cancer in an oncogene is automatically dominant, this cell right here is shown to have cancer due to cancer being dominant and overpowering the other allele on the mom's cell. Now, in tumor suppressor genes, cancer is recessive due to TSG being the reasoning on why cancer will not occur in the cell. So for a cell to be happy, the TSG will have 2 dominant alleles shown here, in which it's expressing that tumor suppressor genes are on and cancer is completely off. If one of the alleles undergo a mutation, so say the mom gene causes a mutation and it turns into little a, this turns off. Once this occurs, it's heterozygous combination. The cell is still known to be healthy due to
TSG being turned on. So since TSG is dominant, it still overpowers the mama allele that is off. So here, Cancer is still not on and is not being expressed. The tumor suppressor genes are. Now, say if another mutation occurs and now it's homozygous recessive, and both genes are shown to be off, this is when Cancer is on and being expressed. So here you have Cancer.